Good evening, everyone. We'll wait for a few more minutes to let others join and then we can start. Hello all, let me just share my screen. Yeah, can you all see this in a slideshow mode?
Yes, ma'am.
Yeah, thank
Yes,
you.
ma'am.
So good evening everyone and welcome back to week 10 of the PMRF TS sessions for the NPTEL online course Drug Delivery Principles and Engineering. So we'll first look at a few of the key points that were discussed in week 10. Yeah, so starting with adaptive immunity. So for the past week, in the last week, we have been introduced with the concept of immunity, and we've been talking about it for a long time now because the reason why we need to learn about immunity is in the context of biomaterials being introduced in a human body. So since these biomaterials are foreign and we have seen that they get involved a lot of in the drug delivery techniques as well as tissue engineering and so on and so forth. So the introduction of these foreign biomaterials in the human body will definitely launch an immune response against these biomaterials. So for that and to avoid that, to make the therapy successful, we first need to know how the immune response gets generated and what are the ways to overcome this immune response or kind of suppress it. So in the past week, we have seen about immunity, and we discussed that there are two branches of immunity. First is the innate and the adaptive immunity. So we talked about innate immunity in the sense that this is the first line of defense whenever any foreign particle gets introduced in the body. So the cells of your innate immunity, such as macrophages or neutrophils, these are they are the first ones that encounter the pathogen. So in case they are not able to eradicate the pathogen from the body, that's where your adaptive immunity will come into the play. So your first response is innate immunity, which is very immediate to the infection, after the infection, and then later on, after a few days, you will see the adaptive immunity kicking in. So we talked about there being a branch in between this innate and adaptive immunity in the presence of antigen presenting cells. So what do these cells do? So these are dendritic cells of macrophages, and they are very phagocytic cells, meaning they'll whatever foreign material they encounter, they'll try to phagocytose, they'll engulf the particle, they will chop down that particle and they'll present it on their membrane, right, for the cells of the adaptive immunity to recognize that this is the particular antigen that you are supposed to fight against. So that's what antigen presenting cells do. And then we talked about the cells of adaptive immunity, which are your B cells, which are responsible for the antibody generation. And then you have a host of T cells wherein they could be cytotoxic T cells, they could be helper T cells, or they could be regulatory T cells. So this was all about the cells of the immunity and how there's an interplay between the innate and adaptive immunity. So in this week's lectures, we got introduced to a point which is known as vaccines. So vaccines, you must all be very familiar with this concept because we all of us have taken several vaccines. We have taken polio vaccines when we were kids. We have recently taken the COVID vaccine few years back. So what exactly are vaccines? So suppose there's a pathogen and you know what that pathogen is, and to avoid getting severe infection when this pathogen enters your body, we take something known as vaccines. So these vaccines could be an inactivated pathogen or it could be part of that pathogen which we deliver into our body. Let our body develop an immune response against that. So this will be the first immune response, and then your body also makes memory cells with this immune response. So later on, when you get the actual infection, your body remembers that I have already encountered this particular antigen and now I know how to fight against it. So it launches a very severe immune response against that pathogen, killing it. So this is why vaccines are very, very effective in preventing a very severe infection from a particular pathogen. So that's the whole point of vaccines. Just a minute. Yeah, so now these vaccines could be of various different types depending upon when they are given or depending upon their content. So there are prophylactic vaccines, which are the most common form of vaccines, which you all are familiar with. So we take these vaccines before the disease occurs. So prophylaxis meaning anything that is done to prevent the disease before we have encountered the disease. So for example, polio vaccine. So we take it when we are kids to avoid getting exposed or to avoid the infection that will happen later on in our lives. So prophylactic vaccines are given even before the disease occurs. On the other hand, there's something known as therapeutic vaccines. So this we take after the disease has occurred. So for example, let's say there's a particular pathogen that has infected a human body, and the patient's body is not able to launch a very strong immune response against that pathogen. It needs some booster vaccines. So that's where your therapeutic vaccines come in play. So this is still in the research phase, and most of the cancer vaccines fall under the category of therapeutic vaccines. So the patient already has the cancer. You're giving a therapeutic vaccine for the body to strengthen its immune response against a particular pathogen. And next, it could be a live attenuated vaccine. What does live attenuated mean? So, as I said, your vaccine could be a pathogen, or it could be a part of the pathogen. So, live attenuated means that you have that particular pathogen that causes the disease. You have somehow modified it to make it less virulent. So, when you expose it, put it into the body, it is going to proliferate. It is a live pathogen, but it will not be able to cause a very severe disease. So it's live attenuated. You have reduced its efficacy to cause a disease. So earlier this was the common form of vaccine given. You can heat treat a particular heat treat meaning you can heat that particular pathogen at a high temperature. You can modify it to reduce its efficacy in causing a disease, and then you can then deliver this as a vaccine. So the disadvantage with this particular vaccine is, in case your heat treatment has not worked properly, there are chances that you could actually deliver a very potent pathogen into the patient's body, and that will then cause a disease. So instead of giving a vaccine, you are actually making the situation even worse. So by delivering a live organism. So in that case, this is a very risky form. But the advantage is, since your organism is now live, although it is attenuated, the body will generate a very strong immune response against this. So you don't have to take booster dosages if you are delivering a live attenuated vaccine. So given once, it will last for a lifetime. So that is one form of vaccines. Then the other is inactivated vaccine. So to avoid this risk of developing a disease while delivering a live attenuated vaccine, we can have an inactivated vaccine. So this means that you kill the organism, and then you deliver the dead parts of that organism in the form of a vaccine. So now, even though the organism is dead, the pathogen is dead, it will not be able to proliferate in the human body, but it still has all its protein and all its structure intact. So your immune response could be generated considering this dead parts as the antigen. So in that case, now since it's an inactivated vaccine, you'll need a lot of booster doses, because this immune response will not be as potent as the live attenuated one. But herein you have eliminated the risk of delivering a live organism, so there is no chance of a disease occurring while delivering an inactivated vaccine. On the other hand, we also have something known as subunit or recombinant vaccine. So it's usually seen that whenever an immune response is launched against a particular pathogen, it is not against the entire organism, but it could be against one particular subunit. Let's say there is a protein on the surface of that particular pathogen. So the immune response is going to be launched against that protein. So what you can do, instead of giving the entire organism as a form of vaccine, you can take that particular sequence of that protein and you can produce that protein on a mass scale in a different bacteria, and then which will then be a recombinant vaccine, and then deliver this particular protein as a vaccine. So now your body is definitely going to remember that since that particular protein is present on the membrane of that or pathogen. So whenever you get an infection from that pathogen, your body memory cells will remember that we have encountered this particular protein and we know how to launch an immune response. So in that way, your subunit or recombinant vaccine can act. And also there are something known as toxoid vaccines, wherein for few diseases we know that the entire pathogen is not harmful, but the pathogen produces a particular toxin which then causes the disease. So instead of having the entire instead of delivering the entire organism as a vaccine, you can take that particular toxin and then develop a vaccine for that toxin, and deliver that toxin itself as a vaccine, and then the body will remember that whenever there is an infection, that pathogen will produce the toxin in your body, and then the immune response can act. So these are the different types of vaccines that were discussed in the lectures. Next is particle-based delivery for vaccines. So now that you know how vaccines are developed, you have to deliver these vaccines. So instead of delivering it as it is, what we can do is we can make micro or nanoparticles of these vaccines. So we learned about micro and nanoparticles few lectures back, right? So that most important advantage of micro and nanoparticles is, since they are of a very small size, your macrophages and your dendritic cells very easily uptake them. So our main aim of delivering vaccine with particles is we want most of the vaccine content, the whatever it is, the live attenuated pathogen, or it could be a subunit pathogen. We want that particular molecule to go into these macrophages, which will then enhance the immune response by antigen presentation. So we want most of these particles to go there. So what we can do, we can develop micro or nano formulations of these vaccines. You take a particular your molecule which is going to act as a vaccine and encapsulate it into a polymer, make microparticles of these, and then deliver. So now your APCs, which are your antigen presenting cells, will have a very higher uptake that will then launch a very strong immune response. Another advantage of delivering vaccines through particles is co-delivery of adjuvants. So adjuvants are nothing but these are molecules which help your immune system to launch a very strong attack. So they enhance the immune system. So along with your antigen, you can also deliver adjuvants because of this particle method. And next advantage of this method is it can carry all types of antigen. So your antigen need not necessarily be a protein. It could be a DNA, it could be a RNA. So all of these could be delivered by making the micro and nanoparticles. And then next point in this week's lectures is immunoisolated cell therapy. So this is something different than the vaccine part. So what is immunoisolated cell therapy? Let's take an example. For example, as I've listed here, let's take the case of pancreatic islet transplantation. So suppose there's a person who has type 1 diabetes. So what happens in this type 1 diabetes is your body considers, so in diabetes, insulin is not produced at a required amount in the body. So in this particular type 1 diabetes, in the normal cell, your insulin is being produced by beta cells of your pancreas. So in people who have type 1 diabetes, it's an autoimmune disease. So the body of that person will consider these beta cells as foreign, and it will start killing its own beta cells. So in that case, your insulin won't be produced. So that's why people get type 1 diabetes. So in that case, what you can do to rectify the situation, you can externally deliver beta cells. Now, if you deliver the patient's own beta cells again and again, the body is still going to think that it is foreign, and it will kill the beta cells. So in that case, what you can do, you can take the beta cells of the patient, and you can encapsulate them in a polymer membrane, and then deliver this particular implant in the patient's body. Now, what this will do, the membrane that you have selected is going to be selective to only few molecules. It will allow nutrients to come in. It will allow the insulin that is produced by the beta cells in the encapsulation to go out, perform its activity. What it will do, it is not going to allow it will create a barrier for your immune system. It will not let your macrophages or dendritic cells or your neutrophils or the antibodies to come near these beta cells. So now what you have done is, by encapsulating these beta cells in a membrane, you have isolated this particular implant from the patient's immune system. So that is why this is known as immunoisolated cell therapy. So it could be used to transplant cells which produce a particular protein, and this polymer layer that is there, it will allow immune isolation and it will protect the particular beta cells. So this is one concept. And the last concept that was discussed in this week's lectures is gene therapy. So you must have heard about gene therapy a lot. So what happens is, we know what genes are, right? These are DNA sequences which code for a particular protein, and that protein then performs a lot of functions in the body. Now let's say there is a person in that particular body, in the patient's body, a particular protein is not being released, not being formed. Now, why this could happen? This could be because the gene that is responsible to produce that protein could be missing, or it could be defective. So it will lead to a defective protein, and then the function of the protein is not happening. So this could lead to a disorder. Now, to rectify this situation, what you can do is you can deliver the gene which is missing in the patient's body. So this is known as gene therapy. You either deliver a gene that is going to produce that protein, or you deliver the correct gene and replace it with the defective gene that is present in the body. So this is gene therapy. So to do that, you can deliver the gene as it is, but then there are a lot of barriers that come up for gene therapy. Some of these could be when you deliver the gene, let's say you make a solution of that gene and you put it through injection in the bloodstream. Now there are a lot of proteins that are present in the plasma. These could then degrade your gene. So the gene is already lost even before it can reach the cells. Even if it can evade some of this degradation and reaches the cell, now your cell is having a cell membrane, right? And your gene is negatively charged. So it cannot enter the cell membrane directly. So for that, you need some amount of help to the gene to enter it into the cell membrane. So this we could do with packing this gene in particular micro or nanoparticles as well. Now, even if it enters the cell, this gene has to reach to the nuclear membrane for it to be transcribed and then translated to a protein. So there are several barriers inside the cell as well. So some of these barriers were discussed in this week's lecture. We'll see some of these in the subsequent questions as well. And in the next lecture, next week, we'll see how to overcome some of these barriers for gene therapy. So these are the points that were discussed in this week's lecture. So with this knowledge, then we can try answering some of the questions of the assignment. Is it okay if I go ahead with the assignment? You can tell me if I'm going too fast or if I'm not audible or clear enough in my speech, so I can repeat some of these things. Okay, so let's look at the first question. The first is, in immune isolated therapy, the cell should be within what distance from the nearest blood vessel to facilitate effective diffusion of nutrients? So I'll give you people some time to read through the options. If anybody knows, you can try answering. If not, we'll discuss. Anyone wants to volunteer for this? No? Okay, then let's look at the answer. The correct answer is 500 micron. So we were just discussing about immune isolated therapy, right? So what do we do in this? We take the cells of that is going to produce your particular protein and we encapsulate it into a polymer membrane. Now this implant we cannot put just anywhere inside the body because this implant doesn't have its own blood vessels. So it has to be near the blood vessels that are already present in the body so that it can get the nutrients as well as, because your nutrients, your oxygen is all going to be transferred from the blood vessel to the implant, because the cells are living inside that implant. They have to get the nutrition, they have to get oxygen, they have to release the waste, and that's how they're living inside. So that implant cannot be very far away from the nearest blood vessel, so it has to be within a distance of 500 microns, so that with passive diffusion the nutrients and your oxygen can then easily pass inside that particular implant and the cells can use it. So if it's more than 500 microns, you're creating a problem for the cells to live. So if you go through the slide, it's clearly mentioned here. So I hope this explanation is clear for this particular question. If there are any doubts, you can ask me right now or at the end of the assignment and we can go over it again. If no, then I'll move on towards the next question. Okay, so the next question is, which of the following would antagonize immune isolated therapy, and we have to select two correct options. So the question is essentially asking us what is disadvantageous if we want to use immune isolated therapy. What are the options from these all two options that are going to lead to failure of immune isolated therapy? So take your time in reading through the options, and if anybody wants to answer, you can. Anyone wants to give it a try? No? Okay, let's look at the answer. So the correct answer is use of biodegradable polymer to encapsulate the cells, or use of a polymer that allows free exchange of proteins and cells to encapsulate therapeutic cells. So we were discussing about immune isolated therapy. So the whole point of immune isolated therapy is to isolate the therapeutic cells, the cells that are going to produce the therapeutic protein, from the patient's immune system. So if you, let's say, have a biodegradable membrane polymer to encapsulate the cells, over the time what is going to happen, this polymer membrane is going to biodegrade, and that will expose your therapeutic cells to the patient's immune system. So this is what we don't want. So that's why one option for this answer is use of biodegradable polymer to encapsulate the cells will actually antagonize the immune isolated therapy. Next is use of a polymer that allows free exchange of proteins and cells. So this is also what we don't want, right? You want to choose a polymer membrane in such a way that it will have small pore size, and it will not allow a free exchange of proteins and cells to come inside. So if it allows the proteins and cells to come, your antibodies, which are very large proteins, they could come in and they could start attacking the therapeutic cells, or your macrophages and dendritic cells could come in and they could start interacting with the therapeutic cells, which is what we don't want. So that's why the two correct options are use of biodegradable membrane and a polymer that allows free exchange of proteins and cells are actually not good, and they will antagonize the immune isolated therapy. Is this clear?
Yes.
Yeah. Thank you. So it's also mentioned in the slides. I have included the slides after each question for you people to read through. So the next question is, the process of weakening a pathogen while generating a vaccine is called what? So we just talked about this in the introductory slide as well. So if anybody wants to volunteer.
Attenuation.
Okay, we have one answer as attenuation. Anybody else? Okay, no other response. Yeah, you're perfectly correct. It's known as attenuation. So the process of attenuation is to basically decrease the efficiency or virulence, make the pathogen weak by either heating it or treating it with some reagents. So this is used in your live attenuated vaccines. Okay, let's look at the next question then. Next is, what are the major challenges of immune isolated cell therapy? We have to choose two correct options. Anyone knows the answer? Wants to give it a try?
Option 4 and 5.
4 is 1, 2, 3, 4. Attacked by CD8 plus T cells, and this approach cannot be used in immunocompromised individual. Okay, thanks for answering. Anybody else has a different response to this question? No? Okay, let's look at the answer. The correct answer is option 1 and 2. Generation of immune reaction and fibrosis of implant over an extended period. So these are the main challenges for an immune isolated cell therapy. Now, why uh, this makes sense is because it could, even after isolating your cells from the immune system, there are chances that it could lead to an immune reaction. Why? Because you must remember the few lectures back we talked about this complement system, right? So these are very small proteins that are present in your plasma, and they are usually flowing around in the body in an inactivated state. So the moment they encounter a pathogen or any biomaterial that's foreign to the body, they will get activated, and in that process they can activate the rest of the immune system. Now these small proteins, they are of a size somewhere around 20 to 25 kDa, and the membrane that you have is ideally supposed to be of a 50 kDa cutoff value. So it means that anything less than 50 kDa size can easily pass through this particular membrane. So this is because you want your therapeutic protein to also go out, you want nutrients to also come in. So in that case, if in case your implant gets contacted with this complement proteins, they can easily enter the isolated cell therapy implant, and they can get activated. In that process, they can launch an immune reaction. So that is one major challenge. Next is fibrosis of implant over an extended period. So this could happen because now your immune system is not able to get rid of this particular implant because it's very big in size, and your immune cells are not able to contact the implant properly because of your polymer membrane. So in that case, what happens, your immune system will then completely wall off this implant from the body. It will completely isolate it, and then fibrosis so a lot of proteins that are present around will start adhering on this particular implant, and it will lead to fibrosis of that implant. In that case, you have to remove it because it's not a very good condition. So these are the major challenges of immune isolated cell therapy. So the rest of the options we'll just go through. Fragmentation of the implant doesn't usually happen. Attack of CD8 plus T cells will not happen because your CD8 plus T cells are very big in size. They will not pass through the 50 kDa cutoff of the membrane. This approach cannot be used in immune compromised individual, which is not true. It could be immune compromised, meaning the immunity of that particular person is not very active. So in that case, you can use this approach as well. And a wide variety of polymers are available to encapsulate cells. This is also true. It's been researched a lot, and we have a lot of polymers to choose from to select for this immune isolated cell therapy. So the major challenges are it could lead to immune reaction, and fibrosis can happen over an extended period. Is this clear?
Yes.
Yeah. Thank you. So let's look at the next question. Next is, which of the following methods can be used for encapsulating cells in biomaterial? We have to choose two correct options. Okay, let's look at the answers. So the two methods which could be used for encapsulating cells in biomaterial is ionic gelation and microfluidics. So ionic gelation is nothing but you have a solution of alginate, and in this particular sodium alginate solution, you can put your cells and then drop it very precisely, one drop by one drop, at a very slow fluoride into a calcium chloride solution. So your calcium chloride and sodium alginate have a tendency to cross-link. So the moment you start adding alginate to a calcium solution, it will because of these calcium ions, it will cross-link. It will form a polymer network. Inside this, your cells could be trapped in this particular encapsulation. So this is one way to encapsulate your cells, and the other is microfluidics, in which you have a precise control of the fluid. So you can then control how much amount of fluid is going in what direction. So these are two methods to encapsulate cells in a biomaterial. Clear? Okay, if there are no doubts, I'll move on towards the next question. Next is, which of the following is a type of therapeutic vaccine? So we have to choose two correct options.
Ma'am, option 2.
Okay, option 2, you're right. Do you have, because we have to choose two options, two correct options, do you have any other answer apart from option 2? Thanks for answering. You're partly correct. Okay, let's look at the next answer. So yeah, you, as I said, you're correct in choosing one option. So we were just discussing, right? Therapeutic vaccines are given when the disease is already occurred, or the pathogen has already been introduced in the patient's body. So it could be cancer vaccine. The other is rabies vaccine. This might be a little confusing, because rabies vaccine, technically speaking, is a prophylactic vaccine. So what happens is, when you get bitten by a rabid animal, the pathogen takes time to travel from the site of bite, from there to the nervous system. So in this interval, if you introduce a particular vaccine, that could prevent the rabies from spreading. So it's technically a post-exposure prophylactic vaccine, but in the context of this question, it could be treated as a therapeutic vaccine as well. So for if you encounter a question like this, which of the following is a type of therapeutic vaccine? It is cancer vaccine and your rabies vaccine. Makes sense? Okay, I'll take the next question for discussion then. Next is, which branch of immunity is triggered when a vaccine is administered to an individual? Again, we have to choose two options from the list. No one? Okay, let's look at the answer.
Passive vaccine.
Sorry. Go ahead. I think I just flashed the answer. So yeah, the correct answer is humoral and adaptive immunity. Sorry for interrupting you. So whenever a vaccine gets delivered, so it is going to be very specific, right? So whatever molecule you are delivering, your uh, immunity is going to be very specific against that. So one is your adaptive immunity is going to get triggered. Humoral immunity is just a part of adaptive immunity. It is nothing but the immunity generated by your B cells, wherein they produce the antibodies. So that's why these two answers are correct for this question. So it's humoral, which is your B cell mediated immune response, and your adaptive immunity. Okay. Let's look at the next question. Next is what is an adjuvant in the context of immune response? So I'll wait for a few minutes for you to read the options and answer.
Option 2.
Okay, option 2 is a molecule that stimulates the immune response to target antigen. Okay, thank you for answering. Anybody else has a different answer from apart from option 2? No one? Okay. Yeah, you're correct. The correct answer is option 2. Adjuvant is a molecule that stimulates the immune response to target that particular antigen. Okay. So the next question is, which of the following are undesirable properties of a polymer used for immune isolated implant? So again, two correct options. So we have been talking about this a lot. So this could be easier to approach. Anybody knows the answer? No? Okay. The answer is toxic and biodegradable. So it's asking us what properties we don't want in the polymer that we are going to use for immunoisolated implant. So as I said, first of all, it shouldn't be toxic. So the entire purpose will be defeated. If the polymer itself is toxic, it will uh, generate another immune response. Rather than generating it for the therapeutic cells, it could be against the polymer, and then your body will start generating immune response against this, and this will lead to a failure of that implant. So it shouldn't be toxic and it shouldn't be biodegradable, because we just discussed in the previous question as well. If it starts degrading, it will eventually expose the therapeutic cells to the immune system, and then this will again your therapeutic cells will be defeated, degraded there, and then the protein will no longer be produced in the body. So yeah, your polymer shouldn't be toxic and it shouldn't be biodegradable. Okay, if there are no doubts, I'll move on towards the last question of the assignment. So this is, which of the following is a potential disadvantage of particle based approaches while delivering vaccine to antigen presenting cells? Again, we have to select two options. Anybody wants to give it a try? No? Okay, let's look at the answers. The first is, so the question is, what could be a disadvantage of delivering particles which are carrying your vaccine to the antigen presenting cells? So first is, the formulation can be unstable because let's say you have not encapsulated the vaccine properly into a polymer. It could lead to disintegration of that particular polymer once you put it in the patient's body. So therein the content of that uh, particle will already be released even before reaching the antigen presenting cells. So in that case, this is not a good approach. So one disadvantage is the formulation can be unstable. Another is the antigen might lose integrity in the process of encapsulation. So we just talked about one method of encapsulating vaccines into particles, right? The gelation method. So in that case, if your protein is not very stable, if it gets exposed to very high concentrations of sodium or calcium ions, it can, the protein will get denatured. So in that case, the antigen will not have potency uh, to activate the immune system. Or in some cases, in few of the methods of making particles, it will be introduced to a lot of solvents which are used in formation of these particles. So in that process, the antigen might lose integrity and it will not be very effective form of vaccine. So these two are the disadvantages of particle based vaccine delivery methods. That's all the questions I have for you for this assignment. So if anybody wants me to discuss any of these questions, wants me to repeat it, or wants me to repeat any of the concepts, we can go over it again. If not, then yeah, that's all I have for you today. We can meet again in the next week. Is there any other problem here? If not, then I'll stop the meeting. You want me to discuss any other concept here, or shall I stop the meeting?
Thank you, ma'am.
Yeah, thank you. Thank you for joining.
Thank you, ma'am.
Thank you.